Hello everyone, today we'll talk about the enzymes and machinery needed for DNA replication. So let's start with it. The very first thing that we need for the DNA replication is the origin of replication. Origin of replication. It is, we also call this as ORI. This is the point in the DNA, this is the region in the DNA at which the replication starts. Let's take an example. The origin of replication is here in the DNA. So this is the point at which the replication starts. The replication starts. Right? Now let's go with the second thing. Second thing is the enzyme which is called DNA helicase. DNA helicase is the enzyme which unwinds the DNA and forms a replication fork. Now what is the meaning of this? So first of all, let's suppose this enzyme binds here and it uncoils the DNA. It unwinds the DNA helical structure. So what will happen? It will open up like this. And it will make a fork-like structure. How fork-like structure? Like this. This is a fork-like structure. So this is what DNA helicase does. Now third enzyme is DNA topoisomerase. This enzyme prevents the supercoiling of DNA. How it does? For example, DNA helicase is working around here and there will be a supercoiling of DNA here. So it prevents this phenomena so that there is no more tension on the DNA. There is no more pressure on the DNA. Right? So next is very important DNA dependent DNA polymerase enzyme. What this enzyme does, it catalyzes the polymerization of DNA. This enzyme is very accurate. This helps in making new nucleotide chains of DNA, and it is highly accurate and it makes around 2000 base pairs per second. It makes a chain of 2000 base pairs in 1 second. Now next enzyme we have is DNA ligase. DNA ligase, this is the enzyme which joins two nucleotide chains or two DNA strands, or we can say DNA strands. It acts as a glue for the DNA because it joins two DNA strands. Now sixth thing that we need for the DNA replication is DNTPs. The full form of this is deoxynucleotide triphosphates. So we have 4 types of these: DATPs, D-adenosine triphosphates, D-thymine triphosphates, cytosine triphosphates, and guanosine triphosphate. So it has 2 functions. First of all, it provides energy for the DNA replication. This is just like ATP. ATP is the energy currency of the cell, and these 4 provide energy for the DNA replication. Now, second point of function is it acts as a substrate. Substrate means it will give ATCG bases for the newly synthesized DNA strands and hence completes the replication of DNA. I hope you understand this topic. If you have any questions, you can leave a comment. Thank you so much for today and have a great day.